Hey guys, my name is Dei-Dei Jimenez, and today we're going to be going over glycolysis and gluconeogenesis, exploring the function and regulation of aldolase. Let's begin. The sum of all chemical reactions in the body that maintain life can be wrapped up in a small phrase known as metabolism. Processes like catabolism, aka the breakdown of molecules to release energy, and anabolism, the use of energy to build cellular components, are included with this. The central metabolic pathways are glycolysis and gluconeogenesis, which manage energy production and glucose homeostasis. More specifically, glycolysis is a process occurring in the cytoplasm where glucose, a 6-carbon sugar, is broken down into 2 3-carbon molecules of pyruvate. Unfortunately, it isn't that simple, as there are 10 enzymatic steps, including key steps like phosphorylation of glucose to glucose 6-phosphate, isomerization to fructose 6-phosphate, and the conversion of fructose 1,6-biphosphate to glyceraldehyde 3-phosphate, ultimately leading to the formation of pyruvate. But don't worry, we'll go more in detail later of each one. On the other hand, we have the complete reverse of glycolysis, which is gluconeogenesis, which allows the body to synthesize glucose from non-carbohydrate precursors like lactate, amino acids, and glycerol. Nonetheless, this pathway is extremely important, especially during fasting or intense exercise when glucose levels need maintaining. The liver and the kidneys are where gluconeogenesis mainly occurs, which involves several bypass reactions to circumvent the irreversible steps of glycolysis. Now, keep in mind that both of these pathways are tightly regulated. Activation of glycolysis occurs by high levels of AMP and fructose 2,6-biphosphate, signaling a need for energy, while gluconeogenesis is stimulated by citrate and inhibited by AMP, reflecting the body's energy status. Now that we are aware of how important glycolysis and gluconeogenesis are in energy production and the maintenance of glucose levels in order to ensure that the body functions efficiently under varying metabolic demands, let's look further into them. We're back to the metabolic pathway that converts glucose to pyruvate, which we already know occurs in the cytoplasm of cells, because we mentioned it earlier before. Let's take a look at this pathway step by step, starting with phosphorylation of glucose, which occurs in order to form glucose 6-phosphate, G6P. So that's this step. So during this step, phosphorylation, I wish I could type it. Phosphorylation occurs here during step 1 to get our glucose 6-phosphate, G6P. Catalyzation of this enzyme occurs through the enzyme hexokinase and requires 1 ATP molecule. The next step is isomerization, which would be here, where the now glucose, our product that we had, I wish I had a better pointer, but it's okay, is then isomerized to fructose 6-phosphate. So now we're going here to F6P. The third step in glycolysis is phosphorylation of the result we got here, our fructose 6-phosphate. So during this step, F6P is phosphorylated to form fructose 1,6-biphosphate, F16BP, by the enzyme phosphofructokinase 1, PFK1, which is a key regulatory step in glycolysis. During this step, 1 molecule of ATP is used. It seems like ATP is used for everything. The fourth step is cleavage, where fructose 1,6-biphosphate is split into 2. So now we're here. It's just so you can have a visual. I included this little diagram. And there is 2 3-carbon molecules, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P. The fifth step in this conversion is when DHAP is readily converted into G3P, resulting in technically now 2 molecules of G3P for the subsequent steps. I know it's a lot, but the diagram, one can see the steps clearly. But let's keep going, don't give up yet. I mean, there's a lot more. The sixth step is oxidation and phosphorylation, where each G3P molecule undergoes oxidation and is then phosphorylated to form 1,3-biphosphate, 1,3-BPG, resulting in 2-NADH. So now we have 2-NADH. Which is then followed by the generation of ATP, where 1,3-BPG is converted to 3-phosphoglycerate, 3PG. The eighth step is where the conversion of pyruvate occurs, as the pathway continues with several more reactions. It is ultimately converted into 3PG into pyruvate. Yay, pyruvate. At the end of glycolysis, from one molecule of glucose, we started with glucose. Let me do the beginning. We started with glucose. There is a net gain of 2 ATP molecules and 2 NADH molecules. Two ATP and 2 NADH molecules. Easy enough, right? Especially with the little diagram. As we know, gluconeogenesis is a metabolic pathway that synthesizes glucose from non-carbohydrate precursors such as lactate, amino acids, and glycerol. During fasting, low-carbohydrate diets, or intense exercise when levels of glucose drop, this process is important. As mentioned earlier, gluconeogenesis is just a reverse of glycolysis, but unfortunately it is not that simple. There are steps in glycolysis that are irreversible, so gluconeogenesis employs bypass reactions to navigate around these steps. The main bypass reactions occur at 3 key points in the glycolytic pathway. So the first key point is a pyruvate to phosphoenol pyruvate, PEP. During this step, there are 2 reactions involved. Pyruvate is first converted to oxaloacetate by pyruvate carboxylase in the mitochondria. After this, oxaloacetate is converted to PEP by phosphoenol pyruvate carboxykinase, PEPCK. The second key point is the fructose 1,6-biphosphate to fructose 6-phosphate. During this time, the reaction is catalyzed by fructose 1,6-biphosphate, which bypasses the phosphofructokinase step in glycolysis. The last key point is the glucose 6-phosphate to glucose. The final step is catalyzed by glucose 6-phosphatase, which bypasses the hexokinase glucokinase step in glycolysis. So essentially, in a perfect world, this just goes the opposite direction, right? Except the key points I am mentioning. The bypass reactions we just went over ensure that gluconeogenesis can efficiently produce glucose even when certain steps of glycolysis cannot simply be reversed. Now let's go over some differences between glycolysis and gluconeogenesis. Both glycolysis and gluconeogenesis are tightly regulated processes that respond to the cellular energy status and metabolic needs. Let's compare the two. The activation of glycolysis occurs by high levels of AMP, which gives indication of low energy availability, resulting in the signaling of the need for ATP production. Stimulation occurs by fructose 2,6-biphosphate by enhancing the activity of phosphofructokinase 1, the key regulatory enzyme in the glycolysis process. On the other hand, gluconeogenesis is stimulated by citrate, which suggests a surplus of energy and substrates, promoting the synthesis of glucose. On the contrary, high levels of AMP inhibit gluconeogenesis, as this shows that the cell requires energy rather than producing it. In terms of cellular locations, glycolysis occurs in the cytoplasm of all cells, making it a ubiquitous process for energy production. Gluconeogenesis, on the other hand, takes place primarily in the liver and kidneys. So I did a little analogy here so y'all can see it. Well, it looks like an analogy, but it's not. It's just the setup. Where glucose production is critical for maintaining blood sugar levels, especially during fasting states. These regulatory mechanisms and cellular locations highlight the intricate balance between energy production and consumption in the body. So that's the past 3 slides. That's what they were important for. Let's continue on. Another thing that was essential in the presentation was the role of aldolase in both pathways. Playing a crucial role in both glycolysis and gluconeogenesis, aldolase is an essential enzyme that facilitates key reactions in both of these metabolic pathways. But that's easier said than done. Let's go a little more in depth. In glycolysis, this enzyme works to catalyze the cleavage of fructose 1,6-biphosphate into 2,3-carbon molecules. These molecules are glyceraldehyde 3-phosphate, G3P, and dihydroxyacetone phosphate. This reaction is necessary in order to continue the pathway of glucose metabolism, which allows for the production of ATP and pyruvate. This reaction can be summarized as followed. And this is, I actually typed it out because it's so hard to use this little marker thingy. Fructose 1,6-biphosphate into glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. This reaction is essentially reversed for gluconeogenesis. In this reaction, aldolase catalyzes the formation of fructose 1,6-biphosphate from the 2,3-carbon intermediates, G3P and DHAP. This is important in the case of synthesizing glucose from non-carbohydrate sources. This allows the body to maintain blood glucose levels during fasting or low carbohydrate intake. Overall, aldolase serves as a key regulatory enzyme that ensures the proper flow of metabolites in both energy-producing and energy-storing pathways. We are now going to dig into the isoforms of aldolase, but just a little bit, because as mentioned before, the role of the aldolase plays in both glycolysis and gluconeogenesis is extremely important. We're going to go into more detail as it exists in 3 isoforms. Those are A, B, and can you guess the last one? And C, each of which has distinct tissue distributions and specialized function. Aldolase A is mainly found in muscle tissue and red blood cells, where it plays a part in energy production during anaerobic conditions. Aldolase B is mainly expressed in the liver, kidney, and intestine. It is important for fructose metabolism and gluconeogenesis, helping the body respond to dietary changes. Aldolase C is predominantly found in the brain and plays a role in energy metabolism in neural tissues, providing support for high energy demands of neuronal activity. Now, the significance of aldolase isoforms lies in their distinct expression patterns and specialized functions across various tissue, which are crucial for adapting to the specific metabolic needs of those tissues. Let's first begin with our first isoform, aldolase A, which is primarily found in muscle tissue and red blood cells, as we mentioned in the slide before. In terms of energy production, this isoform is very important, especially during intense physical activity, as it catalyzes the conversion of fructose 1,6-biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is a key step in glycolysis because it allows for the rapid generation of ATP in muscle cells. Next was aldolase B, which on the other hand is mostly expressed in the liver, kidney, and intestines, as we mentioned before. This isoform has a significant role in carbohydrate metabolism, particularly in the liver, where it regulates fructose metabolism and gluconeogenesis. Aldolase B is essential for maintaining glucose homeostasis, especially during fasting or low carbohydrate diets, because it helps convert non-carbohydrate precursors into glucose, ensuring a steady supply of energy. Lastly, we're going to go over aldolase C, which is normally localized in the brain. This isoform is important for glucose metabolism in neural tissues, supporting the high energy demands of the brain. Aldolase C may also play roles in neuronal signaling and development, highlighting its importance in maintaining brain function and health. The presence of these isoforms in different tissues allows for the efficient regulation of metabolic pathways, ensuring that each organ can meet its unique energy requirements. The specialization of aldolase isoforms reflects the complexity of metabolic regulation in the body, allowing for acceptability to various physiologic demands. At first, I didn't understand why aldolase B was so important, but let's go into the regulation of aldolase B expression in the liver. A crucial enzyme found in the liver is aldolase B. This enzyme is responsible for catalyzing the conversion of fructose 1-phosphate into dihydroxyacetone phosphate and glyceraldehyde. For fructose metabolism, this reaction allows the body to utilize fructose as an energy source efficiently. Now let's look at this a little further into the transcription factors that are involved in regulating aldolase B expression and that play a critical role in the liver's metabolic responses. Hepatocyte nuclear factor 1, HNF1, is essential for enhancing the transcription of aldolase B gene, specifically during periods of high fructose intake. By binding the promoter region, HNF1 increases the enzyme's production, which is vital for effective fructose metabolism. I want to write that one down because these are going to be important. HNF1. Okay, so next there's hepatocyte nuclear factor 3, which they're called the same. The only thing that changes is the number. F3, which works in conjunction with, can you guess, HNF1. Together they amplify the expression of aldolase B. This synergy is especially important when dietary changes occur, ensuring that the liver can adapt to varying levels of fructose. There is also C/EBP, CCAA enhancing binding protein, which plays a role in modulating aldolase B expression during fasting states when there is a high need for glucose. CEBP reduces aldolase B levels and allows the liver to prioritize glucose metabolism over fructose utilization. The last transcription factor we're going to go over is D-binding protein, DBP, which contributes to circadian regulation of aldolase B. It plays a part in the expression of the enzyme based on the time of day, aligning metabolic processes with the body's daily rhythms and energy needs. Together, these transcription factors that we mentioned ensure that aldolase B expression is finely tuned to the liver's metabolic requirements, resulting in the optimized utilization of energy from different substrates. Let's continue on with binding interactions. The aldolase B promoter region includes specific binding sites for transcription factors such as HNF1, HNF3, CEBP, and DBP that we mentioned in the slide before. All these factors play an important role in regulating the expression of aldolase B, depending on the metabolic state of the liver. Now, to get a better understanding, I want to go over a metabolic response sample. In this case, it's a high fructose diet where the presence of HNF1 and HNF3 leads to an increase in aldolase B expression. This is important because it facilitates the metabolism of fructose, allowing for the liver to efficiently process this sugar. On the other hand, during a fasting state, CEBP comes into play and lowers the expression of aldolase B. This change allows the liver to focus on glucose metabolism instead of fructose, aligning with the body's energy needs during fasting. With all this being said, we can see that the coordinated actions of these transcription factors allow for the aldolase B expression to be finely tuned to the liver's metabolic requirements. This regulation is essential for efficient energy utilization and helps the liver adapt to varying dietary conditions. This is the part of the presentation where you open the floor to questions and start having discussions in regards to those questions that the student have. So I'm going to let it roll for just a little bit. Okay. Now I left 2 minutes for questions and discussion. Now let's continue on. We have now reached the end of the presentation. So these are the resources I used in case y'all want to go deeper into them. They are very useful and give a lot of information, so I'd really look into that. Well, you guys, this is our final presentation, so I won't see you guys on the next one, but I hope you enjoyed the presentation.